Proteins assume complex three-dimensional structures that determine the final configuration of the polypeptide. The shape of a protein depends on what are the amino acids forming the polypeptide, not only the content, but what are the sequence of those residues. The shape of the protein will also determine what is the function. To create a three-dimensional protein, we will invest disulfide bonds, hydrogen bonds, hydrophobic attractions, and one new one that is called salt bridge. Cysteine side chains will get oxidized to produce disulfide bond. If the cysteine residues are within the same polypeptides, when they create the cross-link with the disulfide bond, they will make a loop. If the cysteine residues are in two different polypeptides, the link between the two cysteine residues will keep together the two chains as a whole. We have seen hydrogen bonds along the backbone in the discussion of the secondary structure of alpha helices and beta pleated sheets. Some amino acids like asparagine and glutamine can form hydrogen bonds sometimes nearby one another, or in many cases those amino acids can be apart from one another and will contribute in the formation of a three-dimensional structure of a protein, what is called the tertiary structure. For example, a polar amino acid side chain on position number 1 can make a hydrogen bond with one that is nearby, but at the same time, a polar amino acid on position number 5 can make a hydrogen bond with one that is far away on the sequence, like 120. Besides glutamine and asparagine, the acidic amino acids can participate in hydrogen bonding in both ways when they are protonated and when they are with a negative charge. Here we present the hydrogen bonds between the side chain of serine and asparagine and also serine and glutamine. On the first case, we see that asparagine is providing the hydrogen bond donor from the nitrogen. On the second case, serine is the one providing the hydrogen for the hydrogen bond with the carbonyl of glutamine. The importance of this is that amino acids that can have more than one source of hydrogen bond donor or acceptor can be reoriented to accommodate groups in the three-dimensional structure of proteins. Also, the carbonyled or NHs of peptides that are not being used in beta pleated sheets or alpha helixes can also be used to stabilize the three-dimensional structure of a protein. For example, we see here the NH of one polypeptide making a hydrogen bond with the carbonyl, and also the carbonyl of the backbone making a hydrogen bond with the side chain of a tyrosine. Non-polar side chains of amino acids are attracted to one another by London dispersion forces or van der Waals interactions. Non-polar molecules have the tendency to aggregate in the presence of water. This is known as hydrophobic effect. When no polar amino acids cluster together, they create a water-free pocket. Another type of non-covalent interaction important in protein structure is what is called a salt bridge, salt link, or ionic bond. This type of interaction takes place when we have the amino acids that are positively charged at physiological pH, lysine, arginine, or histidine, in proximity with either glutamate or aspartate. They must have opposite charges for the interaction to take place. Also, the amino terminal of the polypeptide and the carboxylate end of the polypeptide can participate in this type of interaction. The tertiary structure of a protein is the overall three-dimensional shape that results from the folding of the protein by use of hydrogen bonds, hydrophobic interaction, and salt link, salt bridges, but the most important contributor is the environment in which that protein is found. Proteins will exclude molecules of water, placing hydrophilic amino acids on the surface and hydrophobic amino acids in the core. Proteins will fold in a distinctive manner that is determined by its primary structure. The primary structure will determine if it will have alpha helices or beta pleated sheets or random coil or loops. We have also the presence, we can see here, of disulfide bonds. The folding of a single polypeptide chain in a three-dimensional space is referred to as its tertiary structure. All the information needed to obtain the shape of the protein is contained in the sequence of the polypeptide. Alpha helices will form whenever it is possible to have a high content of hydrogen bonds between the carbonyl and NH of the peptide bonds every 4 amino acids. Bulky amino acids such as proline are called breakers of the alpha helices, creating turns and twists. That's when we have that the pattern of an alpha helix changes to a formation of a beta pleated sheet. Myoglobin is an example of a globular protein. Those are proteins that are soluble in water. They will place hydrophilic amino acids on the surface and hydrophobic amino acids in the core. Myoglobin is a conjugated protein. Conjugated proteins are those that contain a non-proteic part. The non-proteic part of myoglobin is a heme. It is an organic molecule with the iron center where oxygen binds. This is the structure of a heme that is part of myoglobin and hemoglobin, which is necessary for the transport of oxygen. This heme is connected to the proteic part where we have the polypeptide via only one amino acid that is called the histidine proximal. This is the iron center. This is just a cartoon representing the heme where oxygen is going to bind. When the organic molecule required for function of the protein makes a covalent bond with a polypeptide, this is called a prosthetic group. The heme in myoglobin is a prosthetic group because it's making a covalent bond between the iron part and the histidine. Another oxygen carrier is hemoglobin. myoglobin was monomeric, hemoglobin is tetrameric because it has 4 subunits, myoglobin-like because all of them will have similarity in the shape of the polypeptide, and 4 iron centers, 4 hemes. Two major differences between myoglobin and hemoglobin is that myoglobin carries oxygen to the muscle and hemoglobin carries oxygen from the lungs through the bloodstream. Another difference is that myoglobin is monomeric and hemoglobin is tetrameric, 4 subunits that are stabilized by salt bridges between carboxylate ends and amino ends. Because hemoglobin has subunits, it has primary structure, secondary, tertiary, and quaternary structure. The amino acids on the outside of hemoglobin molecule serve mostly to keep it dissolved in the cytoplasm of a red blood cell. Changes in amino acids may not alter the function of the protein as long as they are hydrophilic or hydrophilic. In sickle cell hemoglobin, glutamate has been replaced by a valine. Replacement of only one amino acid creates a life-threatening disease.